Punnett squares. All right, we briefly touched on this a few weeks back, for those that remember. So a lot of this information you will be familiar with. Some of you have not seen this information yet. So just kind of pay attention to what we do today. All right, so the bell ringer questions today. So let's recall, for those that, like I said, have reviewed genetics or in your ingenuity class have seen some of these terms. Who is Gregor Mendel? So I want you to take a few minutes and kind of reflect on these questions with your partner and just discuss briefly, who is Gregor Mendel? So when you hear the name Gregor Mendel, who is that? All right. What are Punnett squares and why do scientists use them? So what's the purpose of Punnett squares? What is that? And then what is the difference between the terms recessive and dominant? So those three questions I kind of want to take about 2 minutes and reflect on those questions. It's okay if you do not know them. Okay, for those that are unfamiliar with the terms genetics and some of these terminologies. But for those that do know, give me your best answer. Write it on the back of the paper that I gave you earlier with the pre-lab questions on it.
There's 3 questions up there?
It's 3 questions. So who is Gregor Mendel? What are Punnett squares? And why do scientists use them? So that's technically 2 parts to 1 question. And what is the difference between the terms recessive and dominant? If you don't know, that is fine. If you don't have an idea what it is, that is okay. It's not, I'm not judging based on right or wrong right now. I just want to see where you are and what you know. If you don't know, it's okay. Don't know, it's okay. Because we're gonna go over it. Get on back out. Alright, can somebody tell me who Gregor Mendel is? Who is that?
Well, he used to breed the pea plants.
The pea plants, okay, he did. So what do you call that? Like what did he do with those pea plants?
He bred them.
Okay.
That's what it
So he is what kind of scientist is he?
a geneticist.
Geneticist. Okay. All right. Okay. We're gonna revisit that. That's a good answer though. We getting somewhere with that. So what are the Punnett squares and why do scientists use them? So what are the Punnett squares? What are Punnett squares?
Oh, they the non-recessive and recessive alleles.
Alleles. Okay. So you know some of the terminology. Okay. Punnett squares is a chart or squares that scientists use to predict offspring's probability. All right, so what their genotypes will be, their phenotypes, their physical characteristics, those things. Again, we'll review it shortly. What is the difference between the terms recessive and dominant?
I know. Dominant is the one on top.
Okay.
The recessive is like 1 in like 2 or like 1 in 3.
Okay, you said 1 on top and then recessive is 1, 2, 3. Okay.
Something like that.
Dominant. So when you hear the term dominant, what does that mean? Somebody that's on top, above, all right, the one that's stronger, okay, the one that's in control. Okay, recessive, what do you think that will be opposed to dominant? Weaker,
Yeah.
okay, or less prevalent, or if you're thinking of colors, the one that doesn't show as much. Okay, good. Okay, Mendel's conclusion. Okay, so Mendel, father of genetics, he is the one. Let me make sure. So who is Gregor Mendel, father of genetics? All right, he's the first important studies of heredity. So he's the one that kind of created this whole thing. He identified specific traits in a garden of peas, no, you were correct, and studied them from one generation to another. Okay, that's who Mr. Gregor Mendel is. Alright, law of segregation. He came up with two things, y'all. Law of segregation and then law of independent assortment. So law of segregation is when two alleles, and you'll learn a little bit more about alleles, for each trait separate when gametes form. Parents pass one allele from each trait to each offspring. So you get one allele from your parent, your mom, and your dad. Basically, that's what that's saying. Law of independent assortment, genes are formed different, traits are inherited independently of each other. Okay, so we'll elaborate a little bit more on those concepts. What are Punnett squares and why do scientists use them? So Punnett squares, the diagram that you see on the bottom right corner, shows the gene combinations that might result from a genetic cross. So when you cross two parents, what is the potential offspring of those two parents? It's used to calculate the probability of inheriting a particular trait, and we define probability as the chance of a given event will occur. So the chance that something will happen. Okay, I see some terms that's on this board that are in your pre-lab questions, so I hope you're answering those. See, pre-lab questions would be on this sheet right here, as we discussed, that you should be defining some of those terms. And we're not gonna stay long on slides, so make sure you're paying attention. Moving forward.
Hold up. The gene combination.
It's okay. Alright, what is the difference between dominant and recessive? So we said that dominant masks the other trait. So it's the one that takes more, forefront of whatever that trait we're trying to figure out. So whether it's the hair color, the eye color, the trait that shows if present. So represented, when we talk about the lettering and the symbols, the dominant trait is gonna represent that capital letter. Recessive is an organism with a recessive allele for a particular trait, will only exhibit that trait when the dominant allele is not present. So there's no dominant allele, then you'll see that recessive trait. So if you see two lowercase letters in an allele pair, then the recessive trait will show over the dominant trait. Okay? And you'll see the terms again, so if you don't get them all, we'll revisit it again. Dominant and recessive examples. So big T again, straight hair. T, lower T is curly hair, so that's the recessive. The lowercase t is the recessive. So if you see two capital T's, that's two dominant traits, so you're gonna see a person with straight hair. Alright, exemplifying that straight hair trait. You see a capital T and a lowercase t is still a dominant allele present, so the offspring will still have straight hair. The only time the offspring would have curly hair is if you see two lowercase t's or two alleles that are recessive to exemplify that curly hair trait. Okay? That's just an example. Alright, so for further connection, we're gonna look at the short video. We're not gonna play the full video, but there's some questions that I want you to just think about, or some things I want you to think about as you watch this video.
When it comes to citing sources, details matter. Introducing Grammarly's citation.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined
What was Mendel studying? What was the plants he was studying? Pea plants, okay?
a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait.
So the hidden trait is gonna be called what? The recessive trait, alright? And the one that is showing is gonna be considered what? What's opposite from recessive? Dominant, good.
From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical.
Alright, homozygous stand for what? How do we define homozygous? Identical, alright, so the same. Okay, so that's one of your terms that's on your paper. Homozygous is gonna be the same, or alike, or identical.
And what we call a heterozygous pea, when the two alleles are different.
And how do we define heterozygous?
When they different.
When they're different, so you have two different alleles. So you would have a capital allele, a dominant allele, and a recessive allele.
This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
Alright, so Mendel and his peas.
What about the phenotype?
And genotype.
Phenotype, so when we talked about phenotype, they were talking about the actual characteristic that's being displayed. So that's the actual physical characteristic of that offspring. So are they yellow, green? They have green hair, blue hair. Alright, so what you can physically identify. That is the definition of phenotype. Genotype, alright, that's the term on your pre-lab. Mr. Craig should be writing that definition down. Alright, pay attention. So phenotype, we say that's the characteristic, the physical characteristic that you can see displayed by that offspring. Alright, whereas the genotype are the allele grouping. So is it a dominant or recessive? Is it homozygous? Is it heterozygous? So the alleles that are being in question.
Wait, one more time, what you said for the phenotype?
Physical characteristics. There you go. Physical appearance. Good. So transitioning. Based on the brief introductory video and some of the opening questions, y'all, we're going to go over, again, the model of how to model inheritance and how to actually use those Punnett squares. And just learning a little bit about Gregor Mendel's theory. So we're going to briefly go over some of the essential questions. Ooh, so you'll understand how to really understand both of those questions as well. After the end of this lesson, you should be able to tell me I can complete a Punnett square for a monohybrid cross, and then also predict the phenotypic and genotypic percentages or ratios for the monohybrid crosses. So that's just basically telling me, you can be able to tell me what that physical characteristic of that offspring will be. So what is genetics? Alright, what is genetics? Genetics is the scientific study of heredity. Alright? What is a trait? A trait is a specific characteristic that varies from one individual to another. Alright, so we all in here have some similar traits, and some of us have different traits. Some of us have brown hair, some of us have blonde hair, some of us have black hair, okay? Some of us have curly hair, some of us have blue eyes, green eyes. Alright, those are the traits. So those are physical characteristics of a person. What is an allele? Alright, so alleles are different possibilities for a given trait. So what would be examples would be brown eyes, would be dominant trait for a cross. So maybe your mom has brown eyes and you have brown eyes. So that means you took the dominant trait from your mom. You took that dominant allele from your mom. Whereas your dad may have blue eyes. And that's the recessive trait because you didn't pick that, you don't have blue eyes. Does that make sense? Okay. What are genes? Genes are the sequence of DNA that codes the protein and thus determines the trait. So again, genes, you get your genes from generations passed down, your parents, then their parents, alright? Again, you see the term genotype and phenotype. Genotype is the genetic makeup of an organism. So those are the alleles. So the genetics, the genes of that organism. The phenotype are the physical characteristics of how an organism looks. So their color, their hair color, if they're tall or short, okay? Homozygous versus heterozygous. Again, terms used to, say again? Okay, you take it, you got it from home. Yeah, you gotta pay attention, let's go. Okay? And for phenotype, you can just put the physical characteristics. Okay? Homozygous versus heterozygous, again, you look at the terms, homozygous, so look at the prefix of that word. Homo stands for same, like, identical. Okay, so you'll see two identical alleles. Whereas heterozygous, different. Hetero is different, not alike. Alright, opposite. So you'll see two alleles that do not look the same. So a dominant and a recessive allele. Alright. This is the Punnett square. Okay, so we're gonna briefly, now you're gonna use your worksheet that I gave you, and we're gonna briefly review how to actually execute, set up in the Punnett square and how to execute a Punnett square. Okay? So how to complete a Punnett square? So this is also in your Google Classroom, y'all. So if you know we're not able to just stay on a slide too long, you can revisit it to see, to be able to see how to execute it as well. All right. So how to actually complete it? Pay attention. Okay. So you have your So based on these two parents, what do you see? So do you see, what kind of parents do you see? Do you see a homozygous parent? Do you see a heterozygous parent? What do you see? So this parent a dominant trait and a recessive trait. Now, for the parent on the side, what do you see? Homozygous what? Dominant, yes, homozygous dominant, because you see two capital A's. Good. So what you'll do is you'll cross them. Okay, the first thing, you'll cross this dominant A from the first parent with the dominant A from the second parent to get your first offspring in the first box. So you see two capital A's. Okay? Everybody see that? Okay? So then you take the second allele in the first parent and cross it with the first allele in the first parent to get your second offspring. So you see a dominant A and a recessive A. And you do the same thing for the opposite. Okay, you got this top A with the second A, you got two dominant A's, alright? And then you got the top A to the left with the with the capital dominant A, and you get another heterozygous offspring. So steps on how to set up a Punnett square. Again, this is on your worksheet as well. So you're going to draw the 2x2 square. This is called a monohybrid cross. Monohybrid, you're only crossing one trait. One trait. Dihybrid, you will be crossing two traits. So we're just going to focus on one trait. One trait meaning brown eyes, blue eyes. So you're just crossing eye color. Rather than a dihybrid cross, you will be crossing maybe eye color and hair color. Okay, and it's called squares. Identify what the alleles is, so you gotta identify who the parents are, so you can be able to set up the actual cross. Label each row with one of the parents, how they were just labeled, then you cross them. Then you determine the actual genotypic and phenotypic ratios. So in this you have what kind of parents being crossed. Heterozygous parents being crossed. So you got two heterozygous parents. They both have a dominant and a recessive allele, okay? So each parent could give either a dominant Y or a recessive Y to the next generation in its gamete. And a gamete is just another word for an offspring, the egg, okay? There's an equal chance of giving either. So again, they set up their heterozygous parents, one on the top, one on the side, and then they crossed, okay? In this case, the Y that is dominant is gonna exemplify that yellow offspring. So the characteristic that you would see in the offspring would be yellow. Okay? And the recessive trait would be white. So in this, how many dominant or yellow offsprings do we have? And you only look within the four squares. How many yellow offsprings will we have? We would have 3 Y. Why is there only 3 and not 4? You are correct, but can you explain to me why there's 3 and not 4? Good, okay, so in each of the 3, she noticed that there's a dominant trait. There's a dominant allele, okay? So all 3 of these, even if they have a recessive trait, is gonna still be what? Dominant. Because that dominant is going to take or supersede that recessive trait. And then we have one recessive, so 1 white offspring. Everybody understand that? Okay, any questions? Good. Alright, so now you try. Okay, so on your sheet, given the parents, so I already gave you the parents. You got a homozygous dominant parent and a homozygous recessive parent. Okay? The T trait is going to be tall for the dominant trait, and the recessive trait is going to be short. So again, remember, we got to set up our squares. You can go ahead and start working on it. You get to set your squares up. Okay? And then make sure that you put one of the parents. So don't separate the alleles. So don't put a capital T on the top and then one on the side. You got to keep the parent together. Okay? So you put one parent on the top, one parent on the side, and then you cross. I'm gonna give you a couple minutes. I'm gonna give you about 1 or 2 minutes to figure that out. Some of you already done it. And what I want you to do is, I want you to actually give me the percentages. So there's 4 squares. So if we have 4 squares, Tyrone and CJ, I need you all to get to working for me, please. Alright, at least attempt it, try it. Alright, I want you to give me the percentage of what the offspring will be. So there's 4 squares, so that's 25%, right, for each square. So you would say, if there's 2 heterozygous offspring, you would say 50% of the offsprings would be heterozygous plants or tall. Okay? And just write that on the same side.
It's like, it seems like short term, like 100%.
So you say 100%, and they're all got a dominant trait in it, it'll be 100% tall. Yeah. Okay, give you about 1 more minute, and then we'll walk through it together. Okay, so remember you keep the dominant together. So the dominant trait, that's one parent. So you're gonna keep it on top or on the side, but you gotta keep them together. You split each parent. You get what I'm saying? Alright? Let's review it. Alright, so first step is to set the Punnett square up. So remember, you gotta put the 2 parents on their placement. So it doesn't matter where you put them at, y'all, in terms of the parents. So if you put the 2 dominants on the side, it's gonna still give you the same outcome. Okay, just keep them together. You just can't split them. Alright? So the first 2 we're crossing is which ones? Alright, so this top dominant and this first recessive. So what would this first offspring be? A heterozygous, so 1 dominant, 1 recessive. What would the second one be if you're crossing this dominant with this same recessive? Heterozygous again, alright? So then now you're gonna cross these 2 again with both of these, with that last recessive, and you're gonna get what for the last 2? Both heterozygous. So you should have all 4 being heterozygous offsprings. So what would be the phenotypic ratio? Say it again. Tall, but what would be the ratio? So it would be 4 tall offsprings.
That's 100%.
100%. So all the offsprings are gonna be tall. Okay? Alright, so let's try another one. So now, on the back side, you got another example. You got 2 more examples. So now you got a heterozygous parent. So you got a tall parent, a dominant trait and a recessive trait. Okay, and then you got a recessive parent. So again, make sure you keep the parents together. Keep the alleles together. Tyrone, I need you to come on and try for me, please. I know.
I've been up for like 4 days. My eyeballs.
Alright, let's focus. Let's focus. 1, 2, 3. 1, 2, 3. Alright, that means refocus. We gotta refocus. Alright, let me see. Good job. Good. And you gave the percentages. Awesome. Let me see. No? You're not done yet? Okay. Let me see yours. So you got 50% what? Because you got 2 50% now, so 50% what? 50% tall and 50% what? Short. Good. Alright, so here we go, executing. Got a tall T and a lowercase t, so we got a dominant trait and a recessive trait on one parent. Then you got 2 recessive alleles. What would the first offspring be if we cross this dominant and Heterozygous. Alright, so we got a tall offspring. Cross the recessive with the recessive, you got what for that second offspring? Homozygous recessive, so you got a short offspring. Alright, so now we're gonna cross this tall with another recessive, you get a what? A heterozygous? Heterozygous, good, so you got another tall offspring with another recessive and a recessive, what do we get? Homozygous recessive, so you should have what? 50% tall and 50% short. How many of us had that? Raise your hand. How many of us didn't have that? Raise your hand. Okay, so if you didn't have that, tell me where's one way, where did you not correctly place either the alleles, or how did we not get the correct answer? I got you, okay, so you just got confused with the details, okay? Anybody else? Anybody else get anything outside of whoever didn't get 50 and 50? What did you get? Okay, all right, let's move on. Again, should have 2 and 2, 50%, 50% tall, 50% short. So remember, each box is 25%. Anytime that you do a monohybrid cross. Alright. So we will move into our activity. Alright, probability lab. So so if you go back to the beginning of your sheet, on your pre-lab sheet, okay? So go back to the first sheet with the pre-lab questions. Alright, the directions are at the bottom. So each group, you're gonna come and pick up 2 paper bags. 2 paper bags. One bag has a female on it, it says female on it, and then one bag has male on it. Okay, see, one has female, and then one has male. Okay, you're gonna pick up 2. You and your partner are gonna pick up 2 of them. Actually, I'm just going to go ahead and hand them out to you. So I'm gonna give you 2, one of each. Alright, what you're gonna do at the same time, you're gonna have 15 drawings. 15 drawings. So you and your partner are gonna reach down into your bag, either or. So remember, if you got the male bag and the female bag, it's important for you to pay attention to that. Alright, because that's the parent that you are. You're either the male or the female right now. Okay? So, hey, listen up, focus. Alright? So once you all, this one doesn't have anything on it, so this is gonna be the male. Alright? This one doesn't have anything on it, so that's the male. Alright, so what you're gonna do now is you're gonna pull, hey, listen up, 1, 2, 3. All eyes on the screen, let's go. So one student in the group will be in charge of the male bag and the other student will be in charge of the female bag. Alright? At the same time, each student will reach into their bags. Alright, so you're gonna reach into your bag and you're gonna pull out, not right now, right now. No, not right now. Alright, you're gonna reach into your bag and you're gonna pull out a bean, just one. The only possibilities that can be made from the selections are homozygous dominant, so you either have RR, okay, which is red, so the red beans will be considered dominant, and the white beans will be considered the recessive trait. Okay, so in your bag, in each of your bags, you both have 15 of each. 15 red, 15 white. Okay, so depending on which one you pull, that is going to be the allele match. Does that make sense? Okay. So you either have 2. The only possibilities that you can have is you can have a RR. So if you pull a red and she pulls a red, you both have an RR. Okay, so that would be what? Homozygous dominant. You can have R, so you can have a dominant R. So you can have a red bean, then somebody can pull the white bean. So that means it'll be a heterozygous. Or you both can pull 2 white beans, and that will be what? 2 recessive. Okay, so the Y is recessive. Mark your results on the data sheet. So as you pull them, you're gonna mark them on the data sheet. Does that make sense? So, and then you're gonna determine the phenotype of that offspring. So the first row is the genotype, so that's the allele. So again, if you pull in a red, that's a capital R. You pull a white, that's a lowercase r. So you're gonna record that allele in that first row, and then you're gonna give me what is that color of that offspring. Everybody understand what I'm saying? Alright? Ready? You got 15 pull-ins. After you pull, you gotta put the bean back in the bag and re-pull. You can look at it after you pull it. No, when you pull. Ready? Let's go. Make sure you're recording them all. So y'all both got 2 reds, so that's gonna be what? 2 capital what? 2 capital R's. So you will put the 2 capital R's in this row right here, next to the 1, because that's the genotype. And then what color would that offspring be? So the red bean is exemplifying what? A dominant trait, right? So it's R. R is gonna be the dominant trait, and the dominant color is red, correct? So then you got a dominant trait, so it's gonna be what? Red. So phenotype is a physical characteristic, remember? Good. Alright, so y'all gonna do that 14 more times. Okay, which ones did y'all pull? So you got a dominant and you got a recessive. So the dominant bean is a what? Capital what? R. So you got capital R. And then the recessive is what? So you got a white bean, is what? Lowercase r. No, lowercase r. So the offspring, if you look up there, alright, so you got 3 offsprings that you can get. A homozygous dominant, which is going to be red. Heterozygous red, so that means it's an R, a capital R, and a lowercase r. So the recessive trait, the dominant trait is still there, right? It's still present. So the color of this offspring is going to be what? Red. Good. There you go. Hey, it shouldn't be taking you long now. We should be pulling our beans. Come on, let's go. Alright, so, did you pull your bean? Okay, so what's your color? Alright, so you both got white. What is white exemplifying? Is it a dominant or recessive? Recessive, good. So for the recessive trait, it's going to be a what? A lowercase r, right? So if that's the case, the genotype, remember genotype is the allele. So you're going to put 2 lowercase r's, because you both pulled a white bean. And then what color is it going to be? White. You got 2 recessive alleles. The only time it'll be red, hey, listen up. The only time your phenotype should be red is if you have a dominant allele. So if you pulled a red bean. What? So if you pull a red bean with a recessive, so if you pull, anytime you're drawing, so if you pull a red, you pull a right, that's still gonna be red. If you pull 2 reds, it's still gonna be red. The only time it'll be white is if it's 2 white beans. Good. No. So genotype, so what did you pull? A red, what did you pull? Okay, so red is a dominant trait, so that's gonna be R, capital R, and then he had a recessive, so you're gonna put next to that a lowercase r, so you got the recessive. So for the color of it, so if you cross both of them, the dominant trait is present, so it's gonna take prevalent in the color of the actual offspring, so it's red, yes. Only time it's gonna be white is if both of you all pull 2 white beans. Everybody got it? Yes. Go ahead. All right, and as we are doing this, y'all, give you about another minute to work through this. I need you, hey, listen up. Once you have been complete the post lab, the chart. But you can only answer those questions if you completed the chart. Okay? Once you completed the chart, you are done for the day. Okay, your activity, once you have done the chart and the questions. Sorry, the questions, the post lab questions. So you gotta answer the questions based off of what you pulled and what your drawings were. Yes, Cindy. So what is the genotypes of the parents? So the genotypes, just asking you like what genotypes did you come up with? So you don't have to write them all the time. So if you see one, so whatever genotypes that you see, you get ready to put. So you have homozygous dominant one, you have heterozygous one, and you have recessive. So you just put those three.
You sure?
Alright? So as you are working through that, I need you to listen. Alright, so just keep listening. You can work through it, but I need you to listen. Alright, so can somebody tell me who is Gregor Mendel?
The person with the he went.
The person with the P plan, sorry, give me a little bit more.
He figured out like there was different traits and stuff.
He figured out the different traits. He figured out the recessive dominant traits. Good, alright, so the father of genetics. So anytime you hear about Gregor Mendel, oh, that's the father of genetics. He's the one that crossed the P plans to give us a lot more information about how we can predict the probability of an offspring. Good. Genotypes are the alleles, so the alleles that you're crossing. So see, these are the alleles. So you got a recessive, you got a recessive allele parent, you got a dominant, and then you got a heterozygous. Word. Good. So you just put the three different ones that you see. All right, and last question: what are Punnett squares, and why do we need those? Why do scientists use those? Hey, listen up. Listen to the answer. Punnett squares. Good. So scientists use Punnett squares to be able to determine the probability of the offspring. So also to be able to determine what their genotypes, their phenotypes. All right. So we can be able to determine a lot by those Punnett squares. All right. Those were your essential questions today. So repeat after me. I can. Repeat after me. I can. I can. I can. I can tell you who Gregor Mendel is. Can you? Alright? I can use a Punnett square to determine the probability of an offspring. Hey, listen up. Y'all supposed to be repeating after me. I can determine the probability of an offspring by utilizing a monohybrid cross. You just used a monohybrid cross to determine the probability of the offsprings by pulling beans from the bags, all right, and determine their genotypes and their phenotypes. That is the conclusion of our lesson today. Great job, y'all.